We are looking at the endoplasmic reticulum. This consists of the rough endoplasmic reticulum and the smooth endoplasmic reticulum.
The rough ER is a vast system of three-dimensional cavities, some sac-like and some tubular, bounded together by membranes that are located in the cell's cytoplasm. Rough ER transports materials through the cell and produces proteins in sacs called cisternae, which are sent to the Golgi body or inserted into the cell membrane. Proteins made by these ribosomes are transported through the ER to other parts of the cells. The RER isolates and transports proteins once they have been made. Cells in the gut that produce digestive enzymes have large amounts of RER, and adaptation of the rough endoplasmic reticulum is that it has a large surface area for synthesis.
Main functions of the RER is that it is involved in some protein production, protein folding, quality control and dispatch. The rough endoplasmic reticulum gets its name because it is studded on its outer surface with ribosomes. These are called membrane-bound ribosomes and are firmly attached to the outer systolic side of the ER. About 13 million ribosomes are present on the RER, in the average liver cell. Rough ER is found throughout the cell, but its density is higher in the nucleus and the Golgi apparatus. Ribosomes in the rough endoplasmic reticulum are called membrane-bound and are responsible for the assembly of many proteins. This process is called translation. The rough ER, working with membrane-bound ribosomes, takes polypeptides and amino acids from the cystole and continues protein assembly. It is in the rough ER, for example, that 4 polypeptide chains are brought together to form hemoglobin. Protein folding unit. It is in the lumen of the rough ER that proteins are folded to produce highly important biochemical architecture and will provide lock and key and other recognition and linking signs. From the rough ER to the Golgi. In most cases, proteins are transferred to the Golgi apparatus for finishing. They are conveyed in vesicles or possibly directly between the ER and Golgi surfaces. After finishing, they are delivered to specific locations.
The main function of the smooth ER is where the synthesis of lipids such as fats and carbohydrates such as sugars and metabolism takes place, and the detoxification of drugs and other toxins. The smooth ER and the rough ER are separate and are not joined together. The smooth endoplasmic reticulum is involved in the synthesis and transport of fatty molecules such as steroids and lipids. Large amounts of smooth endoplasmic reticulum are found in cells that specialise in lipid metabolism. Cells of the ovaries and testes, which produce the lipid-containing hormones estrogen and testosterone, contain large amounts of the smooth endoplasmic reticulum. The smooth ER synthesises steroid hormones such as aldosterone, testosterone and estrogen.
The ER works closely with the Golgi apparatus, ribosomes, RNA, mRNA and tRNA. It creates a network of membranes found through the whole cell. The ER looks like a flat balloon. Sacs of the ER called cisternae store the complex molecules. It also stores ions in solution that the cell may need at a later time. Steroids are a type of ringed organic molecule used for many purposes in an organism. In liver cells, the smooth ER produces enzymes that help detoxify certain compounds. Liver cells remove alcohol and drugs from the bloodstream. Liver cells have an impressive network of smooth endoplasmic reticulum. In muscles, the smooth ER assists in the contraction of muscle cells, and in brain cells it synthesises male and female hormones. The smooth ER is a tubule network, and the rough ER is a series of flattened sacs. It consists of tubules and vesicles that branch, forming a network. In the smooth endoplasmic reticulum, in muscle cells, the vesicles and tubules serve as a store of calcium which releases as one step in the contraction process. Calcium pumps serve to move the calcium. In the gonads, which is an organ in animals that produces gametes, such as a testis or ovary, the smooth endoplasmic reticulum metabolises the steroids and produces the final steroid hormone. After the side chain of cholesterol is sliced in the mitochondria, the product is passed to the smooth endoplasmic reticulum and further modified. Then it is passed back to the mitochondria for final modifications. The two organelles play a sort of steroid volleyball to produce the steroid hormone.